Okay, well, good. Okay. Okay, so in the first lecture focused on chromosomal disorders. Last week we discussed single gene disorders or Mendelian genetic disorders. Today we're going to talk about polygene or complex disorders. And these disorders are the ones that most of us will have to face both in our personal lives and when you're treating patients. Almost everyone is affected by one or more of the very common disorders. And these include things like diabetes, type 1 or type 2 diabetes. They include obesity, especially in the United States and certain other countries. Asthma, which I have. Let's see, what else? Arthritis, which your parents may have beginnings of right now. These are disorders that affect a large proportion of the population, and almost every one of us will have one or more of these appearing sometime during our lifetime. So understanding how to treat these diseases is something that genetics can actually give important insights into. And so in the future, for example, it may turn out that arthritis is not just one disease, but it's a family of diseases that have different genetic causes. And it may be that how you treat the arthritis or some other complex disorder depends on the genetic background of that person. So the way you choose your drugs to treat the patients will differ depending upon that genetic background. So to understand that, how to do that, how to deal with that information, it's important now to learn some of the basic concepts of genetics. Okay, so today we're going to cover, I'll begin as usual with a short historical background, and then we're going to talk about phenotypes in populations. Last week we talked about phenotypes in pedigrees, in families. And today we're going to shift the emphasis away from individual families to population as a whole. And then we're going to talk about genes in populations, and I'm going to very briefly talk about how we map genes, how we figure out where liability alleles are located within the chromosome. And then at the end I'll go over some complex disorders that illustrate some important points. Okay, so the historical background, we're going to talk about Francis Galton and Ronald Fisher. And Francis Galton was a very prominent scientist in the second half of the 19th century. He is distantly related, a cousin, I think, of Charles Darwin, who, as you know, revolutionized biological thinking by discovering the process by which evolution takes place. And Francis Galton was one of the first people to use statistical methods to quantitate human traits, or to study quantitative traits in humans, and these include things like height, weight, IQ. And what he noticed was that many of these traits were distributed in the population according to this bell-shaped curve, which is more formally called a normal distribution or a Gaussian distribution. Now, I'm not going to go into this in a lot of detail. I included a slide at the end, which I'm not going to talk about this time, that explains more clearly what this Gaussian distribution is. But you can think of it as a bell-shaped distribution, which has a maximum point. It's symmetrical. And what it means is if you're looking at something like height, and you measure height in the population, you'll find that there's an average or mean height that's most commonly found. And then you'll find that less commonly you find people larger or shorter, taller or shorter than that mean height. And if you plot every time you find a certain height, and you plot that for everyone in the population, you tend to get this sort of bell-shaped curve. And it turns out that this bell-shaped curve turns up in many places, both in human genetics and in physics. It was originally discovered as the air curve. So, for example, if you were asked to measure the height of your desk with a ruler, and you made 100 measurements, what you'd find is each time you made a measurement, there'd be a little bit of error in that measurement. And if you plotted all the measurements that you made and counted the number of times you got any particular measurement, you get roughly a bell-shaped curve. So the error in that process is roughly balanced between a little bit too short, a little bit too long, but most of the time you measure something close to the true height of the desk. So when he discovered this was almost like it was sort of like almost like a revolutionary discovery for him, revelationary discovery. You know, why should something like human height fit this error curve? It was something sort of mysterious. And even now, I don't think there's a clear explanation of why this fits, except to say that height, like many other of these complicated traits, depend on many different factors that determine your height. And so if there are a lot of factors, the average person has an average number of those factors that that produce a height, that relate to height. A tall person may have a few extra of those. A person who's shorter may have a few less than those. And then so that if you're a tall person, you are sort of, you have an unusual combination compared to the rest of the population. And so that's probably why these traits like height and weight follow the same kind of curve that you get when you measure the height of a desk. So he was one of the first people to sort of understand this relationship. And he also discovered that when you look at the progeny of parents, the progeny tend to, if you're looking at height, for example, the progeny tend to be, if you have two tall parents, actually the children of those tall parents tend to be a little shorter than the parents. Whereas if you have two short parents, the children tend to be a little larger. And this tendency that he discovered, he named regression to the mean. I'll show you a graph in the next slide to sort of illustrate that more clearly. Now, the other thing that I want to mention about this is in the 19th century, Mendel's work had been published late in the 19th century, but it wasn't well known. And so at the time that Galton was working, people didn't have this idea that genes were responsible for traits and that a gene was something that was inherited and passed on from generation to generation in a sort of non-changing form. Instead, the concept was that whatever was causing a certain trait in parents or in two individuals was sort of blended together in the children. And there wasn't this idea of something discrete being passed from generation to generation. That came a few years later. Now, these are illustrations of what I mean by a normal curve. So this dotted line is the mathematically defined curve distribution. And this is an example of how if you had measured heights of all the people in a population, how the distribution of those heights the distribution that you would find. And to Galton's surprise, this fits a normal distribution pretty closely. Now, if this graph shows what happens when you take the average height of the parents, so you take the average of the mother and the father, and plot that on the x-axis, and then the average height of the children, and you find what Galton found is that you get a curve that looks like this blue line. Now I've drawn in what a 45-degree angle would be for this data. And 45-degree angle would correspond to the fact that if you took the average of the parents and then measured the average of their children, or the child, the average of the child, if there was nothing unusual going on, you'd find that those averages would match each other. So you get a 45-degree angle line. But what you see is that what you actually get is a line that is a little not as steep as 45 degrees. So what that means is that tall parents tend to have shorter children than their average would predict, whereas shorter parents would tend to have taller children than their average would predict. And this is the phenomenon that he discovered and called regression to the mean. And this is a phenomenon that is observed for many, many human traits. Okay. Now, the second really important person that is good to know about is Ronald Fisher, who was active during the beginning of the first half of the 20th century. And this guy was an incredible genius. He made not only very fundamental contributions to the field of statistics, inventing many methods for analyzing data and interpreting that data, but in biology he also is considered to be one of the founders of the so-called modern evolutionary synthesis, where the theories of evolution of Charles Darwin were finally reconciled with the theories of Mendel. And so he's just a very important person both in the field of statistics and in biology. Now, what I want to mention here is one of his earliest contributions was he was able to show how Mendel's concept of discrete factors that are passed on from generation to generation can be used to explain traits that seem to be continuous, like height, for example. And the next slide sort of shows how that can be done. So if you have one gene, one factor, controlling a trait, and this would be the case that you would find for Mendel's traits in plants that are all determined just by one gene, you might find it would produce a distribution like this, where you have say these plants were very small, for example, and you'd get a distribution of 1 to 2 to 1 based on just one factor. But what Fisher showed was that if you have more than one factor determining the trait, you tend to produce a more and more bell-shape-like curve. So for example, with 2 factors, you can generate this kind of distribution. And with 3 factors, you can generate this kind of distribution. And if you had maybe 10 factors all working together, you might be able to get a distribution like this. So this is a very simple idea, but until Fisher showed this, and actually he showed this in a very rigorous mathematical way, it wasn't clear how these discrete factors that Mendel had been studying were related to complex phenotypes that you find in humans, like height and weight or intelligence. So this is a very simple explanation of how those two ways of looking at genetics can be reconciled. Okay, so now we're going to talk about phenotypes in populations. And we're going to talk about familial aggregation of disease. So last week we were looking at pedigrees and showing how traits are passed from parents to children to their children, and showing how that, depending upon whether the trait was a recessive or dominant trait, the pattern changed. Here, though, we're dealing with diseases that have lots of factors, and so it's impossible to trace the effects of individual genes in the same way. And so instead, we have to talk about the sort of just tendency for certain diseases to run in families. So we're going to talk about familial aggregation of disease. Then we're going to talk about how disease risk can be treated as a quantitative trait, sort of modeling after the same way that height can be treated as a quantitative trait, something that can be measured on a continuous scale. Disease risk can be measured on a continuous scale as well, or at least modeled. So, and then I'll just briefly mention some factors that can obscure patterns of inheritance. Okay, so the mathematical way of defining how a disease may aggregate within a family is called relative risk. And this is a real simple ratio of the prevalence of a disease among the relatives of an affected person divided by the prevalence of the disease in the general population. So obviously, if the disease tends to run in the family, you find a higher proportion of the people in that family having the disease than you'd find in the general population. So this would be a number greater than 1 for diseases that run in the family. Now, in addition to just this, there's also a prediction that the prevalence of a disease or risk of having the disease should be proportional to the amount of genes that are shared between the person, the proband, the person who came in with the disease, and its relatives. And one way of calculating that, what percentage are shared, is to write out a square like this, and then calculate the number of alleles that are shared. So this is a little bit complicated, so let me explain this. So here you have a father and a mother, and they have, each has 2 alleles, and they're going to be labeled with individual labels. So this is allele A1, this is allele A2, A3, A4. And then this father can donate either of these to the progeny, right, to his sons and daughters, and that's true for this as well. So, but each son or daughter receives 1 from the father, 1 from the mother. So then, to figure out what's going on, the first thing you do is you say, well, how many different combinations of these alleles are possible? And those are written out here. So this A can be donated in combination with A3, A1, or it could be donated in combination with A4. So those 2 different possibilities are written here. Or A2 could be donated in combination with A3 or with A4, and those are written here. Now, the same combinations can be derived from the mother. So if you look at this, these might be the combinations from the father, possible from the father, these possible from the mother. And then if you ask for this combination here, how many alleles are shared? So this would be one, for example, one sibling might have this combination, where the other one has the other combination. How many alleles would they share? Well, in this case, the alleles are identical. So if the father donated this and the mother to this child, or if the parents donated this combination to this child, and the parents donated this combination to the other child, then these 2 would share 2 alleles at that locus. Okay, and then if the combination between the siblings was this and this, they would share 1 allele, whereas they may also share no alleles whatsoever. So then, to figure out what the average sharing should be is, you count the fraction of possible combinations where there are 2 alleles that are shared. And that's 4 out of 16, so that's 0.25 times 2 shared alleles. The probability of finding 1 shared allele is 8 out of 16, so that's 0.5. And the possibility of having no shared alleles is 1/4, 0.25 times no shared alleles. So if you multiply these together, you find in sum there's an average of 1 shared allele. Now, every person has 2 alleles at any locus, so if there's 1 shared on average, that means on average the siblings share half of their alleles. Okay, and so you can do this kind of calculation then for first degree relatives, which include siblings, for example, and then second degree, which would include grandparents or grandchildren, and then third degree would be like first cousins. You can do the same sort of calculations, and then determine the proportion of alleles that are shared. And so the prediction is then, if genetics is causing the disease, you should see a fall off in the risk of the relatives having that disease as they become more and more distantly related to the proband, to the person who came in with the disease. Is that clear or not? The next slide shows an illustration of how this works. Now, this is a very famous study by Irving Gottesman, who's now at University of Minnesota. And what he showed was that if you look at schizophrenia and the risk of having schizophrenia among different relatives that are related to the person who comes in with schizophrenia, the chance of having schizophrenia or the risk of having schizophrenia is very high, is highest for identical twins, where in theory all the genes are the same, all the alleles are the same, and then falls off as you get further away. So fraternal twins, children, siblings, and parents, are all second-degree relatives, and but they differ so they have less of a risk than identical twins, and then these have less of a risk than compared to relatives that share even less DNA in common, and more so than third-degree relatives. So this sort of trend fits the expectation that the more genes that you have in common with someone who has a disease, the higher the risk is that you have the disease itself. Now, these are not exactly equal to each other, and these may represent environmental differences. So, for example, fraternal twins are like brothers and sisters, or brothers or sisters, but they share the placental environment. So it may be that increases somehow the risk associated with having schizophrenia. And children, of course, share the same environment, whereas parents do not. So some of the differences that you see within this group may reflect environmental differences. But overall, this pattern is consistent with schizophrenia having a genetic, at least partially having a genetic cause. Okay, so then to make this sort of more quantitative, geneticists have developed models for risk of disease where the risk itself is treated like a quantitative trait. And so what you can imagine is that in the general population, there is a certain risk that each of us has. And it's hard to measure, we can't measure that directly, but we postulate that there is such a thing as a risk and that it's distributed in this sort of bell-shaped distribution. And so most of us have sort of an average risk. Some of us have a little bit more, a little less risk. But we don't get the disease unless the risks exceeds a certain threshold. So the frequency of schizophrenia in the general population is about 1% in almost everywhere in the world, between 0.5 and 2, but about 1% on the average. And so the idea is that if you looked at the general population, just by chance, about 1% of the population is past this threshold, and that's why they develop schizophrenia. Okay? So there's not one gene or even two genes that determine whether you get schizophrenia. There's a whole bunch of genes probably, plus environmental factors as well. But in order to get schizophrenia, you have to have just that unlucky combination of factors that put you past this threshold. The idea then is that if you look among families of affected individuals, depending on how closely they're related to the individual, the whole curve shifts. So this now might be the population of second-degree relatives of a person with schizophrenia. And here, what you'd find, if you could measure this hypothetical risk-causing distribution, which you can't, this is just a hypothetical idea. The idea would be that if you could measure that, you'd find that this whole curve has shifted to the right. And in doing so, more people now are past this sort of absolute threshold that determines whether you get the disease or not. So then, if you look among secondary relatives of the proband, you find a higher proportion of those have the disease. So the underlying this is this idea that there are many factors that affect the disease. Some are protective, some are increased risk, and it's sort of the balance between these that determine whether you get the disease or not. Now, all these are sort of just ideas. They're theoretical models, but in many cases, this sort of modeling does fit this disease, the observations about the disease. So this is also from Irving Gottesman's work, and he shows how, for example, depending upon whether you're looking at first-degree relatives or third-degree relatives, the risk for schizophrenia increases because the shift, the population average has shifted for each group. Okay, so that's sort of how disease risk can be treated as a quantitative trait, be thought about in the same way as we think about weight distributions or height distributions. Now, this is just a list of things that can get in the way of trying to understand how genetics, how the risk of developing the disease can be hidden or masked. And one problem is phenocopy. So for example, it may be that diseases that have totally different causes may give the same symptoms. So a good example of that might be a fever. If you considered fever as a disease, it's a symptom, but if you decided we're going to call fever a disease, well, there are lots of different causes for having a fever. You might be a cold, it might be some infection of some internal organ. It could be some types of cancers produce fevers. So in that case, if you were trying to find the genetics the genes that cause fevers, you'd have a problem because you're looking at a collection of diseases instead of a discrete disease. So phenocopy, the idea that different diseases could mask, for example, schizophrenia. It's not clear whether schizophrenia is a single disorder or a collection of disorders. There may actually be maybe 100 different types of schizophrenia with 100 different types of genetic causes. But those are just we're not able to distinguish those yet. So phenocopy is a big concern. You want to make sure if you're looking for the genetic causes of a disease that you're talking about a well-defined entity. And then we talked about last week the problem of penetrance, variable penetrance and expressivity. So again, penetrance refers to the proportion of individuals who actually show any aspects of the disease. It's an all or nothing judgment. And even as we discussed last week, even for Mendelian disorders, where we know the gene that causes the disease, not all people who carry that liability, allele develop the disease. And so that makes it hard to make a relationship between the gene and the disease. And as we saw last week too, the expressivity of the symptoms can be very dramatically from person to person. So these are sort of problems when you're trying to understand the genetic basis of diseases. And then as we talked about last week too, there may be locus heterogeneity. And what this means simply is that different genes may contribute to the disorder. And there can be allelic heterogeneity, so that different mutations within the same gene could cause different phenotypes, for example. And then environmental influences are very big. So, for example, it may be that people who have a certain liability or a certain tendency to develop heart disease might do better in some cultures than other cultures. American fast food culture seems to be particularly bad for heart disease, and just the diet that Americans tend to eat tends to push people over the threshold. So when you're trying to determine the genetic causes of disease, all these extra things have to be taken into account. Okay, so the next thing we're going to talk about are genes in populations. And to do this now, we're up last week when we were talking about how genes may travel, how certain liability alleles or protective alleles may be passed from generation to generation in a fixed manner. We talked about a individual having a particular genetic variant. But now we're going to talk about populations instead. And so the way we think about genetic variants is going to change just a little bit. So instead of talking about a particular variant, we're going to talk about the frequency in the population that you find a liability variant, for example. So instead of saying this person has this liability variant and it has this consequences, we're going to generalize and talk about this population has a certain frequency of that, and therefore the probability of getting a disease is increased, say, in this population, and is decreased in another, but is decreased in another population where it's not as frequent. Okay, so we're going to talk about DNA polymorphisms. We're going to talk about the Hardy-Weinberg equilibrium that shows the relationship between alleles and genotypes. And then I'll mention different ethnic differences in allele frequencies and how that reflects on the probability of developing the disease. And we'll talk a little bit about how genetics can tell us about how the human populations throughout the world have developed. Okay, so we've already talked about, I think we talked about this last time, the single nucleotide polymorphisms and microsatellites and mini-satellites. These are single nucleotide polymorphisms are simply positions in the genome where if you look in the population, you find some people with one sequence here and other people with another sequence there. I'll show this in more detail in the next slide. But what it means is that, for example, not all of us have the same DNA sequence in our DNA. And it turns out that each of us have about over 3 million places in the genome that differ from the canonical or average sequence for the human genome. Each of us carries about 3 million differences. And many of these are these single nucleotide polymorphisms where you might find, for example, you might find a C allele here rather than a T allele, where most people in the world may have this T allele, maybe you and I, and your friend, another friend of yours may have a C there instead. Okay. Now, other polymorphisms are polymorphisms in the number of repeats that you find within microsatellites or mini-satellites. And again, we talked about these when we talked about the structure of the chromosome, but microsatellites are stretches of DNA where short pieces, for example, 1 or 2 or 3 or 4 nucleotides are repeated many times. And it turns out that the number of repeats that you find in the population, people in the population in general, can be different. So you may have 10 repeats where I have 12 repeats and someone else may have 8 repeats. And these are, although these can change by that slippage mechanism that I talked about, over a short number of generations at least, these can be stable and can act as markers for the transmission of a certain piece of DNA from one generation to the next. Another type of repeat that is found are mini-satellite repeats. And these tend to be repeats that involve longer pieces of DNA that are repeated many times. And these, again, can either shrink or become longer. And because of that, if you looked in this room at a certain locus where these repeats are located, some of you may have 5 repeats, others might have 8, others might have 10. And this can be then a marker for that place in the chromosome that distinguishes one person from another person. Okay. This is a slide just to explain a little bit more in detail what SNPs are. So the way to think, an example might be is on this chromosome, one of the chromosomes, you have the sequence is identical to the other chromosome, except for this site right here. And so on one chromosome there might be an AT pair here, but on the other chromosome there's a CG pair. Okay, so the places where the sequence vary like this are called polymorphisms. Well, they're called variants, and they're called polymorphisms if the frequency of the unusual sequence, for example, is found more than 1% of the time in the population. Okay. Now, I just want to, can be sometimes a little confusing because there's a pair here and there's a pair here. And when we talk about the polymorphism, we're talking about a comparison between chromosomes, the sequences on chromosomes. So here, this polymorphism would be called A and versus C here. But equally, it could be called T versus G. Okay? So the polymorphism doesn't involve, this just 2 strands of one chromosome. It's a comparison between chromosomes, and depending upon the way it can be defined either as using this strand versus this strand, or this strand versus this strand. Okay. So right now there's a database where over, I think probably by now, at least 15 million of these sites that have been discovered in the human population are cataloged, and they're given these numbers called RS numbers. So every SNP that's been discovered so far has an RS number and then a number that follows it, and then there's usually information about what the difference in the base pairs are, and then the frequencies with which each of these are found in the population. Okay, so that's a little more information about SNPs. Now, if you imagine this is a chromosome, okay, it's sort of the ideal chromosome, and suppose this one line is actually summarizing the data for two lines that you find here. It may be that you go along the chromosome, and this may be maybe 5 kilobases of chromosome here. Go along the chromosome, and the DNA sequence is exactly the same if you're comparing two chromosomes. So they're exactly the same. And then suddenly you come to a spot where there's a difference in one base pair, and that's a SNP. And then you go along for another 1,000 base pairs or so, and then you find another place where the sequence is different between the two chromosomes. So that's another SNP. And you keep going, you find another one, another one. Well, these alleles, so for example, one person may have an A allele, or one chromosome may have an A allele here, the other chromosome might have a C, and so on down the line. It's possible, because chromosomes undergo recombination, it's possible that all these different combinations of alleles might be found in the population as a whole. So instead of talking about isolated SNPs, you can talk about collections of alleles of those SNPs. So this is A, G, C, T, that combination, and that's called a haplotype. Okay, so different individuals may have different haplotypes at a certain point in the chromosome. And in theory, because in theory recombination could take place between any of these SNPs, every possible combination of these alleles could in theory be found. Now it turns out in actuality that there hasn't been enough time during our evolution for recombination to separate every single one of these from each other. So when you look at the number of haplotypes that you find, it's usually fewer than the theoretical possible one. So the theoretically possible number of haplotypes would be 2 raised to the number of SNPs that you're looking at. So this would be 2 to the 5th power. But in actuality, you tend to find 2 or 3 major haplotypes. And then all the others, the possible ones, are either not detected at all or found at much lower frequencies. And I'll explain in a minute why that's the case again. Okay, so that's SNPs and haplotypes. Now, one of the most important sort of formulas that was derived to explain the behavior of genes in populations is something called the Hardy-Weinberg equilibrium. And what this does is a formula that relates the frequency with which you find certain alleles with the frequency of genotypes that exist in the population. So let's go back here for a second. So this A would be an allele that you might be interested in, and it has a certain frequency. In this case, it's found 0.67% of the population, and the C allele is found at this. Now, since these are on different chromosomes, and different chromosomes can come together randomly, you might ask, well, what's the frequency? If you know these allele frequencies here, what's the probability of finding 2 of these, a person with A at both of these positions instead of one chromosome A and the other C? And then so that person would be, that combination is called a genotype, and that person would be homozygous for this A allele, right? There are 2 copies of that A allele. So what's the probability then of finding a genotype that's AA? And then what's the probability of finding the genotype AC? Okay, and what's the probability of finding the genotype CC? So it turns out there's a very simple way of predicting what those genotype frequency should be from the allele frequencies. And that is summarized in this equation, which was first discovered by David Hardy and a German physician named Weinberg at the early part of the century. And the way this works for actually any number of alleles, but it's the simplest to understand if you talk about SNPs, where there are only two alternative alleles. So here, instead of calling, we're going to call the, there's the A allele, big A allele, little a allele. And if the frequency of that A allele in the population is P, and the frequency of the little a in the population is Q, then since that's all there is, those two frequencies have to add up to 1. You find this or the other, nothing else. And then if you make the assumption that that when you look at the number of sperm or eggs that are produced by the people in that population, that the frequency in those eggs and sperm match the frequency that you find in the people themselves, if you test their blood, for example. Assuming that that's true, and assuming that the mating on the average between individuals is random, well then the probability of a large A allele carrying sperm getting together with a large A allele carrying egg with simply the product of those probabilities, so that's p squared. Okay? And the same holds true for Q. So the probability of this coming together is Q. Now there are two different ways of getting one little a and one big A, and so and if these are equivalent, then they can be counted together. So just by adding up, so 1/4 of the time you get this, 1/4 of the time you get this, half of the time you get this. That simply gives you this equation here, where the sum, of course, is going to be equal to 1. So then these numbers here represent the probability of finding the genotype A, big A, big A. This is the probability of finding the genotype big A, little a. This is the probability of finding the genotype little a, little a. Okay? Now this is so simple that you might think, what's the big deal? But it took geneticists a long time to actually understand this. And it was actually it was discovered independently, but one of the people who discovered it was not even a geneticist, he was a mathematician. And he sort of boasted to the geneticists, like, what is taking so long? This is such a simple idea. Why haven't you guys figured this out? But it turns out, it's so simple, but it turns out that it's a very, very useful principle. And I don't have time today to show you all the applications of this, but just to give you a hint, this formula really holds only under certain conditions. And those conditions are that there's a large population, that no new mutations are being produced that would upset this, that there's no selection against certain genotypes. So for example, what if this genotype was lethal, fatal to the person? Well, you would never find this at all. That you'd never find any Q squared people. So this equation wouldn't hold. And that there's no migration from one place to the other.